Hello, my name is Marje. I currently work in Finnish Meteorological Institute, but the work I'm going to show you is something I did during my recent postdoc in Cornell University. I will show you some results from a computer model I developed together with my colleagues there for simulating the spread of wheatstem rust in global scale, paying special attention to long-range spore transport. Well, wheatstem rust can have a very complex life cycle involving an alternate host and several different kinds of spores. It usually spreads through the Urediniospores in the asexual repeating cycle, so that is the part included in the model. Urediniospores are small enough to be transported in intercontinental scale and are produced in huge quantities. This is a schematic of the model. It consists of atmospheric and land parts. The land part includes parameterizations for spore germination, incubation, and sporulation, taking into account leaf wetness, temperature, and weed phenology, and also overwintering. The atmospheric part of the model simulates the emission of the spores from the field, their transport and deposition, accounting for losses in viability due to UV and temperature. The model is currently coupled to the Community Earth System model that provides all the necessary weather data for it in global scale. CESM as a climate model is computationally pretty expensive to run, requiring use of supercomputers. Next, I will be showing you results from two different applications of this model. The first example is a study of the impact of climate change on wheatstem rust. It was published last year in Environmental Research Letters, and there you can also find a more detailed description of the model. Though in that study we used the model in a simplified setup because of the computational requirements. We assumed constant infection in all fields and analyzed the impacts of the climate change on the different parts of the fungus life cycle separately. I will also show you some preliminary results from the full epidemiological model. The most trivial impact of global warming we found is that by the end of the century, the potential overwintering areas will extend, as seen from the orange areas on the map, significantly, for instance, in the United States, Eastern Europe, and China. Interestingly, some other impacts are conflicting. Warmer temperatures will be suitable for sporulation, and stronger turbulence will enhance the fraction of spores escaping from the crop canopy, leading to more spores emitted from a similarly infected field to participate in long-range atmospheric transport. At the same time, spores deposited onto a wheat plant will find it harder to cause an infection because of the drier conditions and temperatures above optimal germination range. We also looked into probabilities of intercontinental dispersion of the pathogen now and in the future. As the sensitivities of the spore germinability and infectivity to UV and other conditions during the atmospheric transport are quite uncertain, we considered 1 and 3 day transport times as representative of events leading to infection with higher and lower probability. So here you can see how far the spores can travel in these times. Darker shading for 1 day and lighter for 3 day transport. In 1 day, spore exchange is possible between Africa, Europe, and Asia, and also Australia and New Zealand. In 3 days, they can reach pretty far in the downwind direction, potentially crossing an ocean and reaching the next continent in viable state. One interesting connection is, for instance, from Africa to Australia. That specific connection is a very narrow hit, so it is very sensitive to any errors in the atmospheric circulation model, but also to potential future changes in wind patterns. Comparing the future simulation with present, we do see some decline in the viable spore transport probability to Australia, while the frequencies of other ocean crossings mostly increase. You can also notice a strong decrease of transport events around the Mediterranean. There is two reasons for that. Firstly, it gets too hot for sporulation, but also the increasing temperature contrast between sea and land changes the wind patterns, and the stronger sea breezes will make it harder for the spores to cross the seas there. So, if we connect these separate parts of the fungus life cycle to a single epidemiological model, we can follow how the disease spreads in space and time. For instance, we can simulate the annual recolonization of United States or an infection starting in Uganda and gradually colonizing new areas. We do suffer from the lack of good datasets to formally validate the model, but qualitatively, it seems to work reasonably well. However, there are large uncertainties in some of the model parameters. For example, the UV sensitivity of the spores. And comparing the simulations where this sensitivity was varied shows that the results are highly sensitive to this parameter, leading to very different infection levels and spread rates. The model also shows that the speed of the spread of this pathogen depends strongly on the fraction of susceptible wheat and availability of host during off-season, even if such green bridges consist of very small amounts of volunteer wheat. So, in conclusion, the model exists and seems to work reasonably well, and thus it can be applied to study the impacts of climate change or different agricultural practices on the spread of wheat stem rust. But as every modeler always does, I would like to stress the need for more observational data, including field reports of rust presence, airborne spore counts, or experimental data on weather dependencies of all stages of the fungus life cycle. Also, with minor changes, the model could be applied to other similar pathogens, such as other wheat rusts. I would like to thank you all for listening. I will be happy to answer your questions at the end of the session, and you're also welcome to email me with any extra questions you might get. Thank you very much.